Just to introduce ourselves, we are from Dr. Reed's Learning and Adaptive class. I am Maddie Barber.
I'm Meg Berlin.
I'm Sally Gamble.
And I'm Shannon Gordon, and our experiment focused on whether sign tracking contributes to skill learning in math. So our main focus was testing a claim, explaining the surprising results from a study done by Reed, Report, and Light from 2013. And our main question is, does sign tracking link to skill learning in math?
The goal of skill learning is to learn from an instructor and complete the skill when the instructor isn't present, or a transfer of stimulus control. Stimulus control is an environmental event that tells us how to behave in certain situations, like to go at a green light. The word for mastering a skill without a prompt present is called autonomy. Developmentally disabled children tend to need someone to prompt them to do the right skill at the right time. Research into stimulus control can help us try to teach developmentally disabled children to transfer stimulus control. Studies of guiding cues and transfer of stimulus control have also been conducted using animals. This figure shows research conducted in 2013. There were 2 rats in the study, and they both were reinforced for pressing levers in a left-right lever press sequence. First group was the lights group. In this group, there was a light panel over the left lever that guided them to press the left lever, and no panel light over the right lever. When they pressed the left lever, the light went off over the left lever and went on over the right lever to guide them to press the right lever. After this, the conditions reversed, so there was no light over the left lever and a light over the right lever. As you can see, the lever press accuracy dropped below the line of random chance. This shows that the rats were still following the lights, and it followed the lights to press it in an incorrect right-left lever press sequence. In the reverse lights group, it was the exact same conditions as the lights condition, but the lights were reversed. There was no light over the left lever and a light over the right lever. When they pressed the levers, the lights would switch. So as you can see, when the conditions switched and it was the lights condition, there was no drop in lever press accuracy, which suggests that they weren't affected by the lights. So after these results were published, the reviewers proposed that the groups in the lights condition were sign tracking. So sign tracking is when a stimulus, the panel light in this case, causes an animal to approach the light, and the animal associates the stimulus with an appetitive stimulus, which is the food pellet. Sign tracking also triggers a series of species-specific behaviors like salivating and foraging behaviors. So the reverse lights group probably used instrumental conditioning because there was no guiding light, and therefore they may have used trial and error learning. So some evidence for this is when you look at the mean sessions in stable here, it's 12.7, but here it's 26.4. So this suggests that the lights helped the rats press the levers in the correct sequence. In addition, another explanation for the longer sessions in this sequence are that the non-guiding light was distracting to this group. So this group practiced a lot more than the other group, and the result of that is that they practiced the same movement pattern long enough that they learned it, which is called a practice cue. So the current study is a response to the question of whether sign tracking aids in skill learning in rats. And so instead of having a Pavlovian or sign tracking condition and an operant learning condition, we have 2 sign tracking conditions, where one light is stronger than the other, or sign tracking and super sign tracking.
So the baseline condition consisted of the rats pressing the levers in a left-right lever press sequence, following their respective lights. This is the lights condition for the strong group. The left lever has a flashing light over it, and the right lever has a stable light over it. And this is what it looks like until the first response is emitted. So then the lights would switch. The left lever had a stable light, and the right lever had a flashing light. The rats in the strong group pressed the lever under the flashing light. The rats received reinforcement after a correct left-right lever press sequence occurred. If an incorrect sequence occurred, they were given a 3-second timeout. And this is the lights condition for the weak group. The left lever had a stable light over it, and the right lever had a flashing light over it until the first response was emitted. And then they switched as well. The left lever had a flashing light, and the right lever had a stable light. And the rats in the weak group followed the stable light. Once stable criteria was met, the conditions for each group switched. The strong group had to follow the stable light, and the weak group had to follow the flashing light. We then re-exposed the rats to the baseline condition for 10 sessions. The claim hypothesized that the strong group would learn the lever press sequence faster and maintain lever press accuracy longer than the weak group in the baseline condition. The claim also hypothesized that the strong group would perform worse than the weak group in the reverse legs condition.
Instead, we actually found these results. In order to analyze the data, we separated the three conditions into different tests. When we compared the groups for all three conditions, there was no significant effect on groups, which just means that neither group outperformed each other in any condition. There is a point that we were a little bit interested in this point right here. We did testing on it to see if it was significant, but we did find that it wasn't significant, meaning there was not an increase in performance during this time. Going back, this is what we thought our results would look like. At the beginning of the experiment, we believed the weak sign group would significantly outperform the strong sign group during this reverse lights condition. However, neither group outperformed each other, and the results showed that they were actually quite stable with each other. Another important aspect of this study is our accuracy criterion. At the beginning of the experiment, our accuracy criterion was for the rats to have 80% accuracy for about 5 days. Well, it came to the point where we were reaching about 25 sessions and the rats were stabilizing around this 60% here. We eventually decided that they weren't learning anymore or any better, so we said that once they stabilized in accuracy, we were going to switch them to the reverse lights condition. And because we did that, we could not compare the groups in the first baseline condition to see if the strong sign group was actually learning more quickly.
Our hypothesis claimed that the rats in the strong flashing light group would learn the lever press sequence quicker, and that their accuracy in the strong group would be higher than that of the weak group. Our results, however, did not indicate any significant differences between strong and weak group, and no significant differences were found between conditions. Our results indicate that sign tracking not only decreased percent accuracy, but slowed down the rate of learning for rats. Our results did not match up with those of the reviewers who claimed that sign tracking could explain the results of read, report, and lay. They claimed that sign tracking improved percent accuracy and contributed to faster learning. They also claimed that rats who utilize sign tracking show an approach to the lights, but these are a complete contrast to our results. There are a couple of possible explanations as to why our results do not match up with those of the claim. The first of these is that the claim was simply incorrect. None of the predictions from the claim were supported when tested in our study. While the claim was well thought out, our experiment does not comply and could indicate a false claim. The second of these explanations comes from the idea of practice cues and how practice cues interfere with percent accuracy. Our rats completed over 30 sessions in the baseline condition in comparison to the 12 sessions it took rats in the previous study to reach an 80% accuracy. In the experiment, the behavior pattern of moving from the left lever to the right lever to the hopper and repeat had a stronger value than the cue of the lights above each lever. This pattern of movement became their practice cue. Our rats showed autonomy without guiding cues and used the practice cues in the memorization of the pattern. Another important thing to note in our study is the presence, not the position, of the lights. Although our rats completed over 30 sessions in the baseline condition, they never reached a percent accuracy of over 60%. It appeared that the position of the lights above the left and right lever did not affect percent accuracy, but the presence of the light itself led to the decrease in accuracy from 80% to 60%. Overall, the results of our study indicate that sign tracking slows down the rate of learning and decreases percent accuracy.
Some future studies to consider include capping the baseline condition at 12 sessions just from the beginning to see how it influences the results. And another study that we talked about was not a sign tracking study, but rather an overtraining study in an ABC design. And we said that there would be 4 groups. There would be a lights condition, then a reverse lights condition with a left-right lever press sequence target, a lights condition to a reverse lights condition, but with a right-left lever press sequence target, and 2 extended training groups for both of those groups for the overtraining aspect of the study. And then the C aspect of it is that there are no cues in order to measure the autonomy.
We would like to thank Dr. Reed for helping us get our opportunity to present. We'd also like to thank the other students of Psych 300 for being dedicated and not dropping any rats in the study, so we love that. And also the reviewers who made the claim, who made this claim for our study.
Are there any questions?
Yes, sir.
Did you use novice rats that have never been in a lab before, or these were veterans that might have carried over from some previous experiment?
They were naive 5-month-old rats.
Pretty cute too. Yes, ma'am?
Did you see any signs in the rat's behavior, or is there any historical knowledge of, is a flashing light in any way aversive to a rat, where that could be seen as in some way a threatening stimulus that would make them want to stay away from that lever?
It's not necessarily aversive, but we do know that rats don't like the light. It was just in the sense of it was getting their attention. It wasn't necessarily appetitive or aversive. It was just the sole purpose to get them to approach.
That was your intention, but did you see anything in the rat's behavior that suggested that it would have a negative impact on their experience?
Well, they still completed the sequence, they just took them a little bit longer, so it shows that it wasn't aversive enough that they didn't press the levers in the correct sequence, so the reinforcement was, I guess, more appetitive than the aversiveness of the lights.
And since we didn't see a significant difference between the flashing light group and the stable light group, there was no evidence that the flashing was more aversive than the stable.
Any other questions? All right, thank you.